go. All right, biology students. Today I'm going to go over genetics and Punnett Squares. Just do a couple practice problems, more specifically to show you how to get your genotypes and your phenotypes when given word problems. A lot of students I see, like if you give them the genotypes on the sides, it's very easy for them to figure out what the inside of the Punnett square looks like, and then of course like your geno and phenotype. But that's not always the case. Sometimes you got to find your genotype. So we're going to go over a couple practice problems, and we're going to be looking at key words and important things to watch out for so that we know what we're talking about. So the introduction, we are just going to be looking at the results of crossing two individuals can be predicted by making a Punnett square. This thing. At the top of the square, there are two possible alleles. Alleles is an important word, so I'm going to underline that, or you can highlight it, and then I'm just going to write it over here. Remember, alleles are our traits. I'm just going to put trait. And then I also like to think about it as alleles. There are my letters. So I can have a dominant allele, a capital letter, or a recessive allele, a lowercase letter. Okay. From the father are listed. So I have father's genes are up here on my Punnett square. Mother's genes are on the side. It doesn't matter if you flip them, if mom's genes are over here and dad's genes are over here. It's not going to make the Punnett square results different. All right. On one side of the square, there are two possible alleles. There's my word for alleles again, for the mother. So we have father's genes and mother's genes. That's basically what that whole first three sentences are talking about. Within the smallest squares of the grid are the possible offsprings, or where the possible offspring to be written. So what the babies might look like in here. Okay. Monohybrid crosses. So remember the prefix mono means one. So we're looking at one trait with monohybrid crosses. So I'm going to put one trait. All right. One, pattern I see, of the characteristics of pea plants studied by Gregor Mundell. Remember the guy that was really into pea plants and noticed genetics. Sorry, that was the bell saying to change classes. Was the color of pea plants. So our trait is going to be color. Green is dominant. That's important. I'm going to put green, dominant. And yellow is recessive. Okay. So important things I've already gotten written down. I knew what alleles are. Remember, those are our traits, or the letters we're going to be looking at. Mono means one, so we're dealing with just one trait that we're going to be looking at. The trait we're going to be looking at is pea pod color, and we have green as dominant. So that's going to be my dominant allele, or my capital letter. And yellow is recessive, so this is going to be my lowercase letter, or my recessive allele. Make a Punnett square predicting your F1 generation produced by placing pollen from a true breeding. Okay. Green pod plant into a pistil of a true breeding yellow pod plant. There we go. True breeding, this is a phrase that's important. It's a good clue to look at. So true breeding means it's going to be homozygous. So I'm going to put homozygous. Remember, the prefix homo means same. All right, so we have homozygous for a green pod plant. So green is dominant. It says so right up here in my information. And if it's true breeding and homozygous, that means both my traits are going to be the same. Both my alleles are going to be the same. Both of my letters are going to be the same. It's green, so it's dominant. I'm going to put homozygous dominant. I'm just going to abbreviate DOM. And now I get to pick what letters I want to use because it doesn't tell me what letters I have to use. I'm going to pick letters that look different. in the uppercase versus lowercase form. That is a pet peeve of mine. And because we're dealing with the word green, I'm going to pick G's, and they are dominant. So big G, because it is a true breeding green plant, which means it's homozygous dominant, because green is our dominant color. And so I'm going to use 2 capital letters, 2 dominant alleles, 2 dominant traits, big G, big G. Ooh. Okay. So we got one parent down. Let's get the other one down. And a true breeding, haha. So remember that's homozygous again. I'm going to write it down again. Zygous for a yellow. Sorry, that was the bell. Yellow plant. And up here, I have that yellow is recessive. So we're going to do 2 little G's. I'm just going to abbreviate this REC equals little g, 2 recessive alleles, 2 recessive letters, 2 lowercase letters. Okie dokie, artichokes. So we have our letters. And once again, it doesn't really matter, but pollen is the male part of the flower. So I'm going to put big G, homozygous dominant, and little g, homozygous recessive on my Punnett square. And our Punnett square is going to give the genotypic. So genotypic is an important word, or genotype is an important word. And this is going to be what's in my genes or my genetics. Genes/genetics, genetics, there we go. It's also my letters. So what letters I'm going to end up with in my Punnett square. So I'm just going to put letters, just as a little reminder. And then we're going to list all the possible phenotypes, and that's nice, they tell us what phenotype is, physical appearance. So phenotype is a physical appearance, the trait you're actually seeing. So when I go into this beautiful garden of pea pods, I'm looking at 2 different types of pea pods. The phenotype is going to be whether the pea pod is yellow or whether it is green. All right. So now, here's the part that seems to be a little easier for most people, and it's actually crossing the Punnett square. So if you've never done a Punnett square before, prepare for a fun adventure of dragging and dropping letters into boxes. So I drop my capital G and I drag my lowercase g into this box. And this G, this lowercase g, goes into this box. And this capital G goes into this box. And so on and so forth and what have you. Okay? The convention is that the capital letter goes first. So I'm going to put big G, little g, big G, little g, big G, little g, big G, little g. Now, all my offspring, genotypically, the letters inside this box are 100% the same. 100% big G, little g. Okay. Now, before we get into the phenotype, sorry, this is my genotype. I should specify genotype because I'm looking at the letters. All right. So we have a capital G and a lowercase g. These are 2 different letters. Even though they're both G, one is uppercase, one is lowercase, and one stands for the dominant allele and one stands for the recessive allele. And that's important because they mean different things. So the fact that these 2 are different from each other, we're not going to call them homozygous. And remember, homo means same. We're going to call them something different. We're going to call them heterozygous. Heterozygous. And hetero means different. So I take two purebreds, two homozygouses. One's a homozygous dominant because it's green. One is homozygous recessive because it is yellow. I cross them and they are all heterozygous. All right. Now, that's really good for phenotype, but we need, that's really good for genotype. We need to look at our phenotype. So, in order for something to phenotypically express the recessive trait, for a pea pod to be yellow, it needs to have two little g's. It has to. That's the only way that's going to work out. None of our offspring have two recessive alleles, two little g's. So we got a phenotype. 100% green. Oh my goodness, let me write a G like a normal person. There we go. Green. Let me write the word green like a normal person. There we go. 100% of them are green. And this makes sense because the father plant can only give dominant traits. The mother plant can only give recessive traits. And I only need one capital letter, one dominant allele to repress the recessive allele. All right. So we have one Punnett square down. We have all our hints and clues underlined and decoded and our information ready to go. So now we're going to try again, and we're going to switch up our parents. Okay, now we're going to take two individuals from my F1 generation. This is my F1 generation inside the Punnett square. My F1 generations are big G, little g, heterozygous, and big G, little g, heterozygous. They're all the same. So there we go. And then we're going to cross them. So father's genes, I have big G, little g. Mom's genes, same thing, because that's what's up here. I'm going to have big G, little g, heterozygous, one dominant allele, one recessive allele. All right, and then we're going to list the phenotypes or physical appearances for all of them. So phenotype, physical appearance. Okie dokie. And same thing, we're going to drag and drop our G's. We're going to drop that G, drag that G, drop the G, drag the G, drop the G, drag the G, drop and drag. Okay, so the genotype in this square is going to be capital G, a dominant allele, and another capital G, another dominant allele. This capital G is going to get dragged over here, so I have a capital G recessive allele because of this little baby up here. And then I have a capital G and a little g over here. So I have another heterozygous. So far I have a homozygous dominant, big G, big G. I have a heterozygous, big G, little g, or a dominant trait and a recessive trait, a dominant allele and a recessive allele. And then last but not least, my little box over here is going to have little g, which is also homozygous, but it's not homozygous dominant, it's homozygous recessive because that's represented by two lowercase letters. All right, it doesn't ask for the genotype, but what the heck, we already figured it out. Let's just write it out over here. So my genotypes, I have a little bit of a variety this time compared to my first Punnett square. Genotype, there we go. I have 25% big G, a homozygous, let's write that down, homozygous dominant, DOM. I have a 50% heterozygous, and I'm able to figure this out because I just can count them. So 1 out of 4 is 25%. 2 out of 4 is 50%. Right? Think about quarters. heterozygous. And then we got to get to 100%. We have 25 plus 50, so that's 75. And we have 25% left, or 1 out of 4. That's 25%. Little g, or homozygous. Recessive. Remember, the prefix homo means the same, hetero means different. So if you are telling somebody like, okay, I got a genotype of 25% homo, 50% hetero, 25% homozygous, with the homozygous's, you have to specify whether it's dominant or recessive, because phenotypically, it's going to make a difference. Speaking of phenotypes, let's see if we can fit it on this page. So I have my phenotype. Okay, so remember, phenotype is the physical appearance. When you go into the garden, what color are those pea pods going to be? And if you forgot, go right on up here. Green is dominant, so we only need one dominant allele, one dominant trait, one capital letter in order to phenotypically or look green on the outside. And because yellow is recessive, we need 2 recessive alleles, 2 recessive traits, 2 lowercase letters in order for that to be phenotypically expressed in order to see a yellow pea pod. Ooh, all right. So let's finish this up. So we have a, the way I do this, and it's going to be the same whether it is a monohybrid, like these things, a dihybrid when there's 16 boxes, or a trihybrid when there's more than 16 boxes. I just look and see, all right, what boxes have a capital G? Because I know if they have at least one dominant allele, at least one capital G, then they are green, and I don't have to pay attention to it anymore. All right, so here's 1 with a capital G, 2, because it has a capital G there, 3, and this does not have any capital G's, so this is my only yellow one. All the other 3 are green. So I have 75% are green, and we need 100. So 25% are yellow. And I'm able to find that out because the only way that I can express yellow when I plant my pea pods is to have 2 recessive alleles, 2 recessive traits, 2 lowercase letters. And that's just the only one. 1 out of 4 is 25. All right. Well, I hope this was helpful. When you're doing Punnett Squares, doing this little bit of information, underlining and setting aside words and knowing what everything means is so important. These are big words and they have lots of meaning. And if you mess up one thing, I mean, it has kind of like a snowball effect. If I had not read true breeding as homozygous, then, and I put heterozygous, that would have messed up this Punnett square, which would have in turn messed up this Punnett square. So please, please, please take the time and just be aware of what the words are actually telling you. And just go ahead and underline the important ones. Don't be afraid to take the extra few minutes to write out the important information on the side, and it will go a long way. All right, I hope this was helpful, and I will talk to you guys later.